Assalamu alaikum. So in this lecture we'll be talking about cell proliferation, differentiation and death, but the main focus of this lecture will actually be on cell death and the mechanism of cell. So our cells, we have so many cells in our body. We have about 10 to the power of 14 different types of cells, and we have so many differentiated cells. We have about 200 cell types in our body. Now, some of these cells in certain tissues contain stem cells, self-renewing stem cells. These are cells that keep on dividing to a certain degree, and whenever there is cell damage, uh, there is cell renewal. And this is a continuous process for certain types of cells. So for example, you have hematopoietic stem cells that can renew about over 1 billion blood cells that are lost daily. Intestinal epithelial cells are also renewed as well, since many of these cells are sloughed off. They are the differentiated cells. They are removed due to the harsh environment that they stay in. So there are different types of stem cells. Now, many of these cells actually sit in the G0 stage. That is, they are quiescent. Okay, they do not proliferate, but they do proliferate when there is tissue damage. So there is renewal of that tissue, and that includes fibroblasts, endothelial cells that make up neovessels, smooth muscle cells, and specific types of epithelial cells such as those in the liver. Okay. Now, some cells, when they get damaged, they do not get renewed like. So many of these cells undergo what is known as programmed cell death. So they go through a process that is programmed, it's timed. Sometimes it happens due to cell or DNA damage. Okay? Sometimes it just happens due to maturation of cells, such as, again, blood cells. Okay. Now, if cells get damaged, for example, by an assault on DNA, or if cells get infected by viruses, then they go through this process of programmed cell death, and this process is known as apoptosis. Now, apoptosis, by the way, before I get into apoptosis, it's different than necrosis. Necrosis is basically when a cell is damaged due to an acute injury, and this necrosis causes, or this damage causes a damage of the plasma membrane, so things leak in, water and things leak in, and organelles can leak out, and that causes inflammation in the site of necrosis. Now, necrosis is different than apoptosis, the process of programmed cell death, which goes through stepwise process that includes DNA fragmentation, followed by chromatin condensation, and then you have breaking up of the nucleus into small pieces. So there is nuclear fragmentation. Cells then shrink rather than inflate or enlarge like in necrosis, and you have the production of apoptotic bodies. And finally, you have cell death, complete cell death, and this is cleared up by phagocytosis carried out by macrophages. So there are different markers for apoptosis to know if cells go through apoptosis or not. And one of these markers is phosphatidylserine. Now, phosphatidylserine is present in the inner leaflet of the plasma membrane, oriented towards the cytosol. Now, what happens during the process of apoptosis is that phosphatidylserine is flipped, so now it is exposed to the outside of cells. And phosphatidylserine in this case would be recognized by phagocytic cells that carry out phagocytosis of these dying cells. So let's go over the molecular process of apoptosis. Now I'm going to start from the end of the process, which is cell death. Now cell death is induced by proteases known as caspases. What these do is that they cleave and degrade proteins. Now these caspases are activated by cleavage, proteolytic cleavage, by another caspase known as caspase 9. Now caspase 9 gets activated by associating itself with a protein, an adapter protein known as APAF. Now this association between caspase 9 and APAF is facilitated when cytochrome C binds to APAF. So where does cytochrome C come from? It comes from the mitochondria. So what happens is that this cytochrome C is released from the mitochondria. It binds to APAF. It allows APAF to associate with caspase 9. Caspase 9 gets activated. It can then activate caspases, and these caspases can induce apoptosis or cell death. Okay. So what do caspases do? What are their targets? Their targets include an inhibitor of DNase. DNase is the enzyme that cleaves DNA. So there is always this inhibitor binding to DNase, preventing DNA cleavage. Now what caspases do is that they degrade the inhibitor, so DNase now becomes active and it cleaves the chromosomal DNA. Now caspases also degrade nuclear lamins. Now remember, lamins are the nuclear matrix proteins. So when the nuclear matrix is degraded, that causes fragmentation of the nucleus. Now there are other proteins that are targeted or cleaved by caspases, but we'll focus on the first two. So what causes cytochrome C to be released from the mitochondria is actually the action of a family of proteins that belong to collectively to a family known as BCL2 family. Now these proteins belong to three classes of BCL2 according to what they do, and they're either anti-apoptotic proteins or they are pro-apoptotic proteins. So anti-apoptotic proteins are those that inhibit apoptosis, and pro-apoptotic proteins are proteins that induce apoptosis. Now these pro-apoptotic proteins can either be regulatory protein or effector proteins. Effector proteins are those that do a certain action. On the other hand, regulatory proteins regulate the function of the activity of effector proteins. Now, pro-apoptotic proteins in normal cells are inactive, okay? And they are inhibited by interacting with anti-apoptotic regulatory proteins. Okay? So what happens in apoptotic cell is that you have a certain signal that activates the process of apoptosis that would activate pro-apoptotic regulatory proteins. These proteins would then interact with anti-apoptotic regulatory proteins, releasing their inhibition on the pro-apoptotic effector proteins, which do the action. So what we have here is we have these pro-apoptotic regulatory proteins being bound to anti-apoptotic proteins, regulatory proteins. So here's what happens now. We have a cell death signal that would induce the activation of pro-apoptotic effector proteins, such as BAX and BAC. Now these two proteins, BAX and BAC, form oligomer on the outer membrane of the mitochondria. So they form a channel. This channel allows for the efflux of cytochrome C from the mitochondria to the cytosol. Once it's outside, it can interact with a path, which can then interact with caspase 9, activating it. Now, caspase 9 can activate caspases, primarily caspase 3. So it converts pro-caspase 3 to active caspase 3. And then caspase 3 can degrade other proteins, like I mentioned before. Now, there are two pathways for apoptosis. We have an intrinsic pathway and an extrinsic pathway. Now, the intrinsic pathway is basically a factor or an event that is induced from inside of the cell. So if you have DNA damage, if you have viral infection, or if there is deprivation of certain growth factors, that is, the environment is not suitable for cells to go through to stay alive. Now, the extrinsic pathway is something that induces cell death from the outside of the cell. So let's talk first about the intrinsic pathway and how it can be activated. What we have here is, let's say we have DNA damage and this DNA cannot be repaired. This damage is sensed by a protein known as ATM protein. This protein would activate a protein known as p53, either directly or indirectly by activating another protein known as CHK2. Now, this p53 protein is given this name because its molecular weight is 53 kilodalton. So it's protein with P, that is, with a molecular weight of 53 kilodalton. So when p53 is activated by phosphorylation, it can then bind to DNA and it can induce the transcription and synthesis of pro-apoptotic regulatory proteins, regardless of what their names are, POMA and NOXA. And these would then activate, they would work upstream of other proteins like BAC and BAX to form oligomers, release cytochrome C, and so on. Now, there's also an external signal that induces survival. And this signal is activated, or it goes through the PI3 kinase AKT signaling pathway. Now, we talked about this pathway in the previous lecture. Now, what happens here is that in the presence of a growth factor, you will have induction of or activation of a receptor. This would cause activation of PI3 kinase. PI3 kinase would phosphorylate a lipid molecule associated with the plasma membrane known as PIP2, converting it to PIP3. So it adds a phosphate to the molecule. Now, this PIP3 would then associate with and activate a protein known as mTOR. mTOR can then activate AKT. AKT, once activated, it can have several actions. For example, it can phosphorylate a pro-apoptotic protein known as BAD. When BAD is phosphorylated by AKT, it would not be active anymore. So what AKT does is that it inhibits apoptosis and it induces survival. Now, AKT also acts on other proteins such as p53, NF-kappa B, and we talked about NF-kappa B before, GSK3 inhibiting it, and GSK3 actually is an inhibitor of an anti-apoptotic regulatory protein. So by inhibiting GSK, you would have this activated. So you would have the action of preventing apoptosis from happening. Or you'll have activation of the mTOR protein as well. Now, this protein is associated with survival as well. Now, there's also an extrinsic pathway that induces apoptosis. So, two slides before, earlier, we talked about the intrinsic pathway. Here, we're going to talk about the extrinsic pathway that induces cell death. And this is activated by a growth factor known as TNF, or tumor necrosis factor. Tumor necrosis factor binds to its receptor, activating it, resulting in the activation of a caspase known as caspase 8. Caspase 8 can activate effector caspases, or it can activate a protein known as BID. Now, BID is a pro-apoptotic protein that would activate BAX and BAC. These two would form the oligomer channel, resulting in the release of cytochrome C, activation of APAF, and activation of caspase 9 and caspase 3, and so on. So what happens here is that this pathway, by the way, the TNF pathway, is actually activated due to activation of the immune system. So these cells, immune cells, would activate, would induce cell death if immune cells suspect that these cells are cancer cells, or if they are virus infected cells. Now, the same pathway is used to eliminate excess lymphocytes at the end of an immune response. Now, there is an alternative to apoptosis, and that is autophagy. We talked about autophagy before. Autophagy is basically cell eating. So the cell eats itself. And this autophagy is actually mediated by the mTOR signaling pathway, and we talked about this pathway before. So what happens is that when cells are under stress, if the environment is hostile, what would happen is that if they have lack of nutrients, what they would do is that they would degrade macromolecules inside them, including organelles like mitochondria, releasing the macromolecules to be used as a source of energy. So basically, this is the fate of cells. So you have a cell in here. The cell can undergo proliferation in normal cases. Now, for stem cells, they keep on going through self-renewal. Now, some of these stem cells, by the way, they stay quiescent until the tissue is damaged, or when there's a signal that tells stem cells to differentiate. So these cells can undergo differentiation and maturation. And that would include fibroblasts, endothelial cells, smooth muscle cells, epithelial cells, and we already talked about that in the first slide. Now, these cells can stay in the state of quiescence. That is, they're active, metabolically active, but they're not growing. But if there is a certain signal, such as if there is tissue damage, they can go through the proliferation, like I said before. Now, some cells go through senescence. This is a case where cells are asleep. That is, they do nothing. There is hardly any metabolism, as if they are dead, but they're not dead actually. Now, these senescent cells can eventually would go through apoptosis, or cells can go through apoptosis directly. So these are the different fates of cells.